So we started the class last day itself while discussing the epigenetic modifications that are visible in the genetic material. Now, it is a recapitulation of what I said the previous day, that epigenetic modification can take place in the early embryo during the formation of gametes followed by zygote. During the zygote, erased, and then the blastocyst stage is arrived. The cells of the inner mass become pluripotent and the epigenetic marks are re-established. Then epigenetic reprogramming is done during gametogenesis in the sperm and the eggs. Epigenetic program is done for formation of constitutive heterochromatin, for X chromosome imprinting, and for position effect heterochromatinization. Now, there are two things that should be very clear in the mind at the onset, that the DNA is wrapped around the histone molecules. So when we talk about epigenetic modification, it includes the modification of the histone proteins and the DNA. For a given program to occur, modifications can take place both in the histone moieties and the DNA jointly, or the modification may take place only in the histone moieties, or the modification may take place in the DNA. So it's a permutation combination, and depending on whether both the modifications are taking place in both the molecules, that is a histone and the DNA, or only in DNA or only in histone, the output is different. So the basic aim of epigenetic modification is either you make the chromatin open. So in the open state, the promoter elements, the enhancer elements, that is the regulatory elements of the genes are exposed, and the different transcription factors or the different proteins can come and interact with the DNA. And in the closed configuration, as you can see, the DNA becomes inaccessible to any outward protein that can DNA binding protein. So that is the very basic thing we should keep in mind. Now, as I had already shown you the picture the other day, that the DNA is wrapped around the histone. Roughly, the histone consists of 146 base pair of DNA wrapped around an octamer. The octamer consists of 8 molecules of histone. There are normally 2 molecules each of histone, H2A, H2B, H3, and H4. In some cases, some nucleosomes contains variant histones such as H2AX, H2AZ, H3.3, or otherwise known as CEN-PA, which is found in the centromeric region. And as you can see from this picture, you can see from this picture that the N-terminal tails of the histone molecules are protruding out from the core nucleosome and are accessible to external proteins. So it is these portions that are accessible to the external proteins. The common modification of this N-terminal ends include the acetylation, the mono, di, or trimethylation of lysines. Other important modifications includes the phosphorylation of serines of histone 3 at the residue 10 and 28, the mono, di, or trimethylation of arginines of histone 3 at the residue 2, 8, 17, and of histone 4 at the residue 3, phosphorylation of threonines at the residue 3, 6, and 11 of histone 3, and ubiquitination of lysines at the residue of histone 2A, 119, and histone 2B, 120. So these are some of the signals that are given when we observe certain kinds of modifications. To read a modification, for example, if I'm reading this modification, you see it's histone 3 K is for lysine. The symbol for lysine is K. The symbol for arginine is R. The symbol for serine is S. The symbol for threonine is T. So when I read a modification like this, so this is histone 3. For the fourth amino acid from the terminal end, that is lysine. So it's lysine at the fourth position, having 2 methyl groups. Me2 means 2 methyl groups. This is histone 3. Lysine at the fourth position. Me3 means 3 methyl groups. So these are various modifications that are observed, and depending on the modifications, you can see these are the messages that are read. So if we are having a modification of histone 3 at lysine 4, either it is a dimethylation or trimethylation, it indicates that the promoter of a given gene is active. So these are the various modifications observed, and also are discussed that there are enzymes which are responsible for modifying the histone molecules or the DNA molecules. So as you can see by the name, lysine methyl transferase. These enzymes add methyl groups to a given residue found on the histone tails. Histone acetyl transferase. These groups add the acetyl groups to the histone molecules. Histone phosphokinase. These groups add a phosphate molecule to the histone amino acid residues. So writers are basically enzymes which add molecules to the histone tail, thereby bringing in a functional output. Erasers are simply those enzymes which remove those molecules that were written by the writers. So if a lysine methyl transferase adds a methyl group, then lysine demethylase can remove that methyl group. Similarly, if there is a histone acetyl transferase adding an acetyl group to a lysine molecule, then histone deacetylase simply removes the acetyl group from the amino acid residues. Apart from this, there is a third category of molecule known as readers. These belong to the class of ATP-dependent chromatin remodelers. What they do, they can, these are big protein complexes, they can identify the modifications on the histone molecules and accordingly can bind with the histone molecules. So they can read only when the writer enzyme has modified some portion of the histone molecule, histone tails. Now coming to this picture, you can see that this is a histone molecule. So this is H3 and this is the amino terminal tail of H3, that's the histone 3, and you can see the various modifications that are being observed. You see the colors of the modifications. For example, the purple, the violet color showing acetylation. The turquoise color is showing deacetylase. Just let me enlarge this picture. So as I was saying, the turquoise color is showing the deacetylase molecules, and then the brown color is showing methyl transferase. So this is how the tail comes out, and in the amino terminal, the various residues are modified in various ways. R represents arginine, K represents lysine. Lysine is a preferred amino acid to be modified because, as I said earlier, the lysine and similarly arginine both have a second amino group. So they have 2 positive charges in them. So modifying lysine would thus nullify one positive charge, thereby reducing the electrostatic interaction between the DNA and the histone molecule. So lysine, arginine, these are preferred molecules. Lysine is the most preferred amino acid for modification. Now you can see this is histone 4, histone 2A, histone 2B. And these are the tails of histone 4, histone 2A, 2B. And again, you can see various modifications that are observed. So given the picture, you can assume that you can at least gauge how much amount of modifications can take place within the histone tails. Now, if we look into the structure, the molecular structure of lysine, so this is the epsilon amino acid. Now, this epsilon amino acid can be modified. A single methyl acetyl group has been attached, or a methyl group can be attached. Just remember, lysine is a positively charged molecule. So what happens when lysine is present in the amino terminal tail? It actually neutralizes the charge of DNA. DNA is negatively charged and histone is positively charged. So this positive charge to some extent is provided by the lysine molecules of the histone moieties. Now, when we add a acetyl group, we are reducing the charge of a lysine molecule by 1 unit. If we add a second molecule of acetyl, we'll also be reducing the charge by another unit. So remember, the moment we are adding an acetyl or a methyl group to the lysine molecule, the positive charge of the molecule is being reduced by a unit. If it's one molecule by unit, if it is two molecule by 2 units, so this is how the charges, positive charges of the lysine gets reduced, and as a result, the histone tends to become negatively charged. DNA itself is negatively charged, and as a result of this negative interaction, the DNA tends to open up from the histone core. So these are the acetyl modification, methyl, dimethyl, trimethyl. And as I showed you in the previous slide, you can see the H3 modifications, the various positions where the histone on the amino terminal tail where histone 3 is modified, and the positions in the tail of histone 4 where the modifications are seen. Just remember, from the amino terminal tail, we do the countdown. So as you can see, this is position 2, S for serine, this is position 1. So the countdown is always done from the amino terminal tail. And this is the lysine acetylation. As you can see, this is acetyl lysine. This is another picture. This is a picture from Bruce Albert's monomethyl lysine, dimethyl lysine, trimethyl lysine. This is the serine modification. We are having phosphoserine, the serine phosphorylation. As I said again, another picture of the various modifications being shown here. And I have told you earlier that there are the writer enzymes and the eraser enzymes. So all these modifications are being done by the writer enzymes. And if they are removed, then they will be done by the eraser enzyme. And the modification state, as I started with that, it actually gives an output signal. So if it's a trimethylation, then the signal is gene silencing. If it's at position 9, lysine 9, again, if it is a methylation at position 4 and the acetylation at position 9 is gene expression. Just remember, addition of acetylation usually favors gene expression, because the moment we are adding acetyl groups, acetyl itself is negatively charged, so the charge of the lysine residue decreases by a basic law I said. So acetylation usually favors gene expression or the opening up of the chromatin. Again, we have another modification shown here, lysine methylation. So at position 27, this is trimethylation. You can see this is a repressive complex found in Drosophila, and these are the variants of the same histone molecule. This histone 3 variants are shown here. Same histone 3 modified in 3 different ways, giving 3 output signals. And coming to the enzymatic reaction, so this is the protein. This is acetyl transferases. So they are adding acetyl. So this is the protein. This is the amino group, and As from the acetyl-CoA, the acetyl group is added to the amino group. And so this is a writer enzyme. And as I said, there are eraser enzymes. The eraser enzymes of the deacetylase remove the acetyl group, bringing the lysine residue into its original configuration. And I wanted to show you a picture of a gel electrophoresis in which it is shown, which reflects the fact that the acetylated histones can be isolated very easily because there is a charge difference. If you remember, when I said that it is monoacetylated, then you have to assume that the residue has a 1 unit less positive charge. Diacetylate, 2 units less positive charge. Triacetylated, 3 units less positive charge. And tetraacetylated, 4 units less positive charge. So if it so be, that is, a tetraacetylate molecules tends to be more negative than the monoacetylated form. So when we resolve them in gel, you can see the this is the histone 4, and this is the monoacetylated form, which is more positively charged as compared to the tetraacetylated form. It being more positively charged tends to be attracted more towards the negative end. So it has been seen that the acetylated forms of histone can be easily resolved in gel because the charges differ on the acetyl molecules. Now coming to the histone variants, we all know that the histone is made up of H2A, 2B, 3 and 4. But apart from that, in our genome, there are certain variations of the histone 2A and the histone 3 molecule. So you see, this is basically all histones have a structural orientation. This is known as the histone fold, you can see. So that is common for all histone molecules. And then the histone variants the H3 has 2 variants, H3.3 and SEN-P. You can see there are small variations in the histone fold. And the H2A has again 2 variants, 2AX, 2AZ. If you look into the output, this variant has a different functional output, and the SEN-P variant has a different functional output. The SEN-P variant is associated with the centromere function and the kinetochore assembly, while the 2X variant has a role in DNA repair and recombination, and the 2AZ variant has a role in gene expression and chromosome segregation. Usually, the histone variants are inserted into the nucleosomes at specific sites on chromosomes by ATP-dependent chromatin remodeling enzymes that act in concert with histone chaperones. So when the replication machinery is taking place, these things actually happen. And I told the variants and their function, and we now move on to the next part. So this is a summarization of the histone lysine methylation part. This is a histone molecule. And depending on the structural modifications, you see that the same histone molecule has different functions. This is the polycomb region. PRC is the polycomb. PC is the polycomb group. So this is a polycomb 1. So you can see that simply by the modification of so this is a modification of histone 4 at lysine 20 methylation, histone 3 lysine 9 methylation, histone 3 27 lysine methylation. So you can see it's the same histone. There are 5 different methylations of the histone 3 molecule. We are getting 5 different output signals. And there is one histone 4 modification, and we are getting another output signal. And this is how, this is also a summary of how both methylation, acetylation together make up a signal. So you can see this is histone 2B. How you read it? Histone 2B. Lysine 123. So is the position of the lysine is at 123rd position from the amino acid tail. And there is ubiquitination. So ubiquitin molecules have been added, which is followed by H3K79 methylation, followed by histone 3 lysine 4 methylation, followed by histone 3 lysine 9 acetylation, followed by histone 3 arginine 17 methylation, followed by histone 4 lysine 16 acetylation, and the output is an active chromatin molecule. So you can see it's a group of it's a mixture of methylation, acetylation together, at times phosphorylation, at times ubiquitination. Together they are giving an output signal. Similarly, a different set of run through of signals gives us a repressed state. So this is just the summary of how histone modifications usually activate or suppress a chromatin area. Now, these are the two techniques. I asked you people to go through the techniques, check through the animations. And if you have any problems regarding this, we can take it up later on. Now coming to the another part of our topic, that is the DNA methylation. So we'll be moving on to DNA methylation. We have done the histone acetylation part. Now we'll be doing the DNA methylation.